Hi, my name is Heather Hanslick, and I'm really excited to present some data from my research on the species Impatiens capensis, commonly known as orange jewelweed. I initially had a snail species in mind for this study, but due to some safety issues, I changed study areas, and I was floored by the plant biodiversity in my study plots and became really interested in finding out how the species varied in each location, why, and what I could find in common. I conducted my research in two study plots on an undeveloped portion of a farm in New England, Georgia. I chose my first study plot along a 30-meter transect line of marsh terrain, as you can see here, and my second study was approximately 20 meters away in an adjacent creek area, also along a 30-meter transect line. Both of these areas receive their water from portions of Lookout Creek. And you can kind of see this city is located in the county of Dade. This is Dade County, Georgia, and it borders Tennessee and Alabama. That's right here in the corner. And then if you kind of zoom in on that creek system, this is where that water is coming from. I used the Picture This Plant Identifier application to aid in identifying plant species and noted an abundance of orange jewelweed in both locations. I then used a 23.5 centimeter by 4.29 centimeter quadrant to form a sample study from each transect line, only to find orange jewelweed as the species in common between the two sample studies. Let's see here. So I was really impressed by those findings and the biodiversity present in each small sample study. I calculated each sample's Shannon index, evenness, and species richness, and you can see those results here. The Creek Area Study Plot 2 sample yielded the greatest species richness. However, the Marsh Area and Study Plot 1 sample had more species evenness and a higher Shannon index, indicating a higher diversity of species. And you can see that there are some, multiple counts of 1 here. And there's a few counts of 1 here as well, but we've got 73 and 13, 18, 10, and 12, where we've got like 103, 1, 1, 1, and 1, and 2. And so that's going to affect that index quite a bit. So based on what I had visualized, that it looked like orange jewelweed, despite competition, would thrive more abundantly in the marsh area than the creek. This sample study was informative, but not determining, and so I needed more data. I saw less competition for orange jewelweed in the creek area of study plot 2, but lots of considerations were in play. The surrounding plant life was completely different in both areas, with far more shade in the creek area of study plot 2. Orange jewelweed will start competing for space with broadleaf cattails, but will also attract hundreds of hummingbirds that prefer to eat and pollinate orange jewelweed in open areas, such as the marsh in study plot 1. So if you can kind of visualize what that looks like, we have a really heavily wooded creek area, a lot of shade, a lot of ground cover, and then we've got a more open marsh area. We're getting more sun. The plant life is different, and the animals that are going to come to the area are different. Going to have a lot of hummingbirds in the marsh area because they really like those open areas. Orange jewelweed grows stronger with hydration and also disperses through explosive dehiscence. These characteristics were indicative of the marsh of study plot 1 having conditions more suitable to sustain life for orange jewelweed. Not only would the plants likely grow stronger and attract more pollinators, they would have more opportunity to disperse their seed. I conducted further analysis of the two study plots by taking one random sample study along the 30-meter transect line of each study plot. Only species of orange jewelweed were selected from each sample, 45 species from the marsh study sample and 40 species from the creek study sample. A measuring tape was used to determine the mean length in centimeters of a twin serrated leaf from each of the 85 species. The measurements were taken from the top of the stem down the center of the leaf to the tip of the leaf. I conducted a statistical analysis of these measurements, and let's see here. My confidence intervals did not overlap, indicating that the mean lengths of the leaves from the two study samples were significantly different. This supported my hypothesis that orange jewelweed would thrive more abundantly in the marsh area despite competition. Though, as you can see here, more variance in leaf length was detected in the creek study sample, and the creek study sample is on this right side here. This is study plot 1, which is the marsh area. You can see the sample sizes, the mean leaf length, variance, standard deviation, standard error, and of course confidence limits for both of those. Yeah, more variance in leaf length was detected in the creek study sample, indicating that explosive seed dispersal was not very successful, possibly due to the number of leaves covering the ground. Explosive seed dispersal looked successful in the marsh area study sample, with an abundance of orange jewelweed species of similar size in close proximity of one another. I predict that by the end of spring, large, healthy orange jewelweed will be filling the marsh area of the study plot 1 and have a weaker presence in the creek area of study plot 2. I'd like to go back to these images of the two study areas and talk about some of the ecological factors I observed that may influence the difference in abundance, and ground cover is certainly one of them. This is the creek area, and you can see how thick the leaves are here. And these plants aren't, even though it is really wet, it's really moist. That's something that this plant has to have to thrive. It isn't as wet as the marsh area, and we can see how these plants are growing in similar size and abundance over here in the marsh. A heavy presence of fallen leaves. The hardiness is different. You can kind of take a look. Of course, this is one small sample again. I would like to take more samples. You can see a difference. In fact, there are some plants that just had a really significant difference, almost like they look like they had some deformities. Their leaves were a lot smaller than they should have been. You can see this looks like a really healthy sample. So perhaps sunlight isn't penetrating the plants in the creek study area as well as it is in the marsh study area. These plants were probably dispersed last year. It definitely wasn't a recent dispersal. This is new growth this year. So they've had the same opportune time to grow. And so they've showed quite a variance for having that same opportunity. I'm interested in repeating this study again in the following year during the same season and taking more sample plots from each study. And to reiterate, though, initially I wasn't sure what to expect in terms of the abundance of orange jewelweed in the two study plots. I was observing large quantities of orange jewelweed in each study plot, and as I identified the surrounding species, I realized how competitive orange jewelweed was, being the only similarity of plant species that I had identified in the two study samples. 19 other plant species were identified in the first study sample of study plot 1 in the marsh, and 34 plant species were identified in the first study sample of study plot 2 in the creek area. So despite abundance at either study plot, the fittest species will attract more pollinators, and the mean leaf analysis indicated that the study sample from study plot 1 in the marsh had fitter species. Here are my references, and I hope you enjoy this presentation. I thank you so much for your time.